Namaskar and welcome to India Science. On this very special telecast today, we are joined today by a very special guest, Professor K. VijayRaghavan, Principal Scientific Advisor to the GOI and the co-chair of the R&D Vaccine Task Force. Always a pleasure to have you speak to us, sir. Thank you so much for sparing your time today. So the vaccine has brought a host of questions to all a billion plus Indians. Cheers and smiles in the new year. And I would start by beginning to ask you, sir, if you can tell us a little more about what a vaccine is and how are these vaccines developed, sir, primarily?
Yeah, thank you very much. This is a very great pleasure to be here interacting with everyone. There are many diseases which are preventable by vaccines. And vaccines typically used to be, pathogens which are inactivated. For example, for viral diseases, a less active virus or an inactivated virus was taken and used to stimulate the immune response, so that when an actual infection takes place, there is a response ready and can deal with the infection. So the earliest vaccines were attenuated vaccines, vaccines which had a lower activity and didn't cause the disease. Viruses, for example, didn't cause the disease. And then when the disease actually was presented, the vaccine was effective. So smallpox, for example, was that kind. Later on, vaccines became of other kinds. People took, for example, viruses and inactivated them. And when that was injected, the body, when it broke up those components, saw those and raised an immune response to it, so that when an infection then comes, the body is ready. Much later, and this has happened very rapidly, there were other kinds of vaccines made in which you make viral components in what is called a vector. And these vectored vaccines use genetic engineering to insert into them components which code for components of the virus. And then they are injected, they go into the body, they make those viral components, and then there's an immune response. And more recently, and quite strikingly, there are vaccines that just inject the viral genetic material encoded in a special way, so that for a special component of the virus, so that an immune response can be raised. So there are many kinds of vaccine platforms now, and the pandemic actually has resulted in the speedy deployment of many of these platforms.
Absolutely, sir. So as India battles this pandemic, so there has been a lot of talk about the Swadeshi vaccine. So has India developed a vaccine earlier? If so, which are these?
The Indian vaccine effort dates back to many years ago. In early the 20th century, for example, when vaccines were made at what were called the Pasteur Institutes, one in the Nilgiris and one in the north, in Kasauli. And these institutes were important sources of vaccine production. The Hafkian Institute was also set up around the early 20th century, and Hafkian came to India, and he was there during a major epidemic and set up the vaccine production, and later on the institute was named after him. Then much later, after independence, Indian vaccine industries have steadily been building up, and we have many vaccine companies making vaccines for typhoid, for Japanese encephalitis virus, and rotavirus most famously very recently. There is a meningitis vaccine which was developed some years ago, and that has been administered to, I think, about 250 million people across the world, mainly in Africa, saved an enormous number of lives. And 2 out of 3 vaccines in global child immunization programs are made in India. So India has got an extraordinary success in vaccine manufacturing and development over many decades.
Absolutely, sir. And जैसे आपने कहा, ये भारत की सतरंगी सभ्यता को भी दर्शाते हैं. From the Nilgiris to all over, we have as a scientific fraternity really come out victorious. So my next question is, the Google charts are really going virus, finding out what a vaccine is actually composed of. So I would like you to tell us, especially the COVID-19 vaccine, what is there in the vaccine, sir?
As I said earlier, there are many, many platforms which are used to make the components which stimulate the immune system through a vaccine. So, if, for example, you take the messenger RNA vaccines, they encode components of the virus, and when injected, they make those components in our body. And once they make that, our immune system attacks it. So, to answer your question, they would be the messenger RNA, as it's called, along with what's called a particle, so that it can be transported. The messenger RNA is stable. Now, messenger RNAs typically are not very stable at room temperature, so they have to be transported very carefully. And mRNA vaccines, therefore, have a very major cold chain requirement. The other type of vaccines are vaccines which are like the AstraZeneca Serum vaccine, which insert a component of the virus's coding sequence into another vector, and that's inserted, and then the body synthesizes that, similar to the messenger RNA vaccine. The Bharat Biotech vaccine, Covaxin, in collaboration with ICMR, takes the virus, grows it up in a laboratory, inactivates it completely to ensure that it's not infected, and then uses that. So not only one component, but many components of the virus's proteins are stimulating the immune system. So there are many, many components. One important point, the vaccine also has other components in it to make sure it is stable, and it also stimulates the immune system. So these are all other things which are added. So it's not just simply being able to make these components, which many labs can do, but it's to make them in very carefully monitored facilities so that they meet the highest medical practice standards and are safe to use.
Yes, sir. We're getting a lot of questions on the hashtag, but maybe we could do that a little later. A question that is coming to us, flashing on Twitter also, is that if the vaccine has the pathogen, is there any chance that I would get the disease should I take the vaccine?
In the set of vaccines against COVID-19, none of them have the pathogen, as it were. You have inactivated virus, but the pathogen is completely inactivated, or you have components. So the vaccines will not cause the disease. In earlier times, many, many years ago, there used to be most vaccines were attenuated vaccines. So they were active, but at a very, very low level. In some situations, they can cause disease or mutate to cause disease. And those were the dangers earlier. That's not the danger in the vaccines which we're looking at now.
Sir, and if I ask you, how are the two vaccines which have been approved by the GOI different, sir? Could you elaborate on that?
Yes. This is a matter which is very important to understand, and important to understand in terms of risk-benefit ratio, as it were. We must remember that vaccines are given to healthy people, and therefore they have to be very, very safe. They also have to be effective. There's no point in giving a vaccine which is not effective. And these are processes which are very, very long. And therefore, the measures of safety, immunogenicity, what kind of immune response you get, and efficacy are part of multiple steps in trial stages. Safety is measured in the stage in phase 1, but it's also measured in phase 2 and phase 3, and also after introduction in post-licensure, post-marketing surveillance. So safety goes across all the time, and it's constantly monitored in different ways, in different contexts, in different numbers. Immunogenicity is measured by looking, after you administer the vaccine, whether there's a response, even in the absence of the disease being presented. So that gives you an idea that the vaccine perhaps is very likely to work. Now, you then have a situation where you have trials where you have the vaccine administered to a set of people, and another set getting the placebo, and both are exposed to the disease. And you ask how many people in the placebo arm are protected against the advancement of the disease, and how many compared to those in the vaccine arm. So these are the kinds of stages which goes through. Now, in terms of the risks which are there in not administering a vaccine, those also come into play. Is the virus so dangerous that you have to compress the stages or speed up the trials because you have to save lives? Is the spread so rapidly happening that you would like to have the trials administered simultaneous to having the vaccines introduced? Those are the kinds of demands. Now, today, with the SARS coronavirus 2, we are seeing an extraordinary surge in the in the expression of certain mutant strains, for example, in the UK. So let me take the example of the strain in the UK that grew in November in their lockdown with very tight non-pharmaceutical interventions, whereas the other strains, decreased in the November lockdown. So the R value, as it is called, for the other strains became less than 1, but this particular strain went higher than 1. So it's very worrisome, and it caused an expansion. And the seeding of this strain was extensive across England in October and November. And then there's a 2-month time lag, which we've seen, that the seeding goes through to extraordinary large numbers. So this is the kind of problems you face, which you have to deal with in deciding whether and when to introduce vaccines. Now, again, it's a bit long, but this is important. If you look at the 7-day rolling average of deaths, of new deaths in India, it is now about 18 deaths per 10 crores as a daily average taken on a 7-day rolling basis. If you look at the UK, it is about 1,013 compared to 18 per 10 crores on a 7-day average. And in the US, it is 808. In other words, it's not, people talk about as if the strain is a problem. The strain itself still is very likely to allow vaccines to be effective. That's not the issue. But the spread which the strain causes is a cause for serious worry. And therefore, it is important to have vaccines in your toolkit which you're able to use. And that's the idea behind the emergency use authorization anywhere in the world. These are the kinds of risks of not vaccinating which are measured. Now, our regulator has given 2 kinds of approvals for 2 vaccines, the Covishield and the Covaxin. For the Covaxin, our regulator has said that taking into account global phase 3 data and some bridging data from India, that an emergency use authorization can be given, and that authorization will have certain conditions of monitoring. And for the Covaxin, there will be, again, an emergency use authorization, but they will have different kind of monitoring until phase 3 results are also examined. So that's the kind of situation which has been done. And therefore, the efficacy measurement for the Covaxin is based on an expectation that phase 3 trials will show that, and an extrapolation of what the efficacy is in non-human primate models. And therefore, that's why a different kind of emergency use authorization are given. Both vaccines are safe. Both companies are extraordinary in what they've done. And I have no doubt that the safety and immunogenicity of the vaccines are strong. And I also have no doubt that as the results come in for both vaccines, as for other vaccines under emergency use all over the world, appropriate ratcheting of the decisions will take place.
Right, sir. Very broad perspective there. Thank you so much. I think it's been wonderful the way you explained the whole thing, and our viewers will get the exact picture. Another question which has been coming in, I think I can club 2 questions with your permission. How will the COVID-19 vaccine protect me from getting infected as one? And we go to the next one, and why are they booster doses, sir?
Right. So as I said, by all laboratory tests and clinical trial tests, it seems that the vaccines do protect against progression of the disease. So that's the good news over there. And vaccines, you must remember, don't protect people against infection. They prevent, upon infection, the progression of the disease. So we must remember that there is a requirement for us to continue to wear masks and keep distance until the virus is reduced in the population, because there will be transmission until everyone or a very large number of people are vaccinated. So that's an important point to keep in mind. So vaccines certainly do protect. What was the second part of your question? Sorry.
The booster doses, sir.
Yeah, booster doses. Yes. So booster doses are necessary because the first dose primes the immune system and gets it ready, and that in some kinds of vaccines, in some situations, would require an additional dose to boost up the immune system so that it is now at a level where it's ready for the infection. In other kinds of vaccines, a single dose is often enough, but the measures of immunogenicity, dosing, will decide whether one or two doses are enough. And this can vary.
Sir, the next question that I have is, can I not continue wearing a mask to prevent getting the disease? I think so, you've elaborated enough. One has to wear a mask, and social distancing is the norm of the day. So, if you'd like to add something to this one also.
Yeah, so I presume the question is that if you are vaccinated, should you still wear a mask?
Yes.
Uh, yes, you
Carry on.
should until the entire or most of the community is vaccinated, so that people who are vaccinated don't transmit the disease to others who are not vaccinated, although they themselves are protected.
So people are also writing in to find out how long will this protection by COVID-19 vaccine last?
There seems to be good news on that, based on studies of how long the immunogenicity, the immune response to infection lasts. And more studies are showing that upon infection, the immune system remembers for quite a while. The range starts from 8 months to a year, but it could well be longer. And there's reason to think that vaccines would offer protection at least in that range, if not more.
So, another question that most people want to know is your take on, will the vaccine have any side effects, sir?
Well, all our lives is full of side effects. Whether you take an aspirin or any medicine, there is always the possibility of side effects. And those side effects fall into multiple kinds of categories. Those immediately after taking the vaccination, which could be minor aches and pains, they usually are not causes for worry. And there are others which potentially could show an impact in a slightly longer time frame. So typically, as I said earlier, the safety protocols involve not just at the early stages of phase 1 or 2 or 3, but they go on in a post-marketing licensure, looking at larger and larger populations, because less and less frequent side effects or effects, adverse events after immunization show up on that scale. Usually, when vaccines are introduced, this monitoring, the early stage trials are such that the side effects will be for the entire population very minor. There are some situations where care needs to be taken. For example, those who are immune compromised, but that is usually adjusted before the vaccination itself.
Another question which is coming to my mind, and I'm sure most of our viewers also want to know, is that what is a clinical trial? If you could please elaborate.
Yeah. In the early days of the development of vaccines, there were little or no trials. Sometimes vaccine developers tried these candidates on themselves or on their family members or friends. Now, over time, this has become standardized in a rigorous way, which is very good. And vaccine development, therefore, involves multiple processes which require permission. And that should be, something which all of us need to understand. First of all, when you make a vaccine candidate, even for testing, you need to make it under rigorously certified conditions so that you can then say it's a potential vaccine candidate. In other words, you just can't make something in the lab and say, here is my vaccine candidate. It has certain good laboratory practice standards which need to be done. Then those are taken and they are studied in cell culture, and those again are very rigorous. These are all preclinical steps, by the way, to make sure that the vaccine candidate meets standards, and then you go through animal trials again, which are rigorously monitored. Then you go to the clinical trial phase. In other words, when you try candidates on humans, they've already passed through a set of trials. The phase 1 trial typically involves a relatively small number of people. Could be varies with the disease and with the vaccine. It could be from tens to hundreds, and you can have, you're basically testing whether the vaccine is safe, checking the dosing and everything, so that you know that what doses are okay and how safe the vaccine is. Then you go to the next stage, the phase 2, where you check the immune response in addition. And this is in a larger number of people. And this can, again, depending on the nature of the disease and the vaccine, be into several hundreds or thousands. And then you go to phase 3, where you go to many more thousands of people, where you actually expose the vaccinated to the disease compared to those who are not vaccinated and see what happens. So this is a long time, and it takes, permissions have to be given at every stage, data has to be shown at every stage, and this typically takes several years, if not decades, for this program to go through. And during the pandemic, this has gone through very speedily because the risks of not vaccinating are so large.
So, which is it safe to participate in a clinical trial? Would you like to say something to this one, sir?
Of course, people have already participated, and you have people all over the world which has participated. The reason it's called a trial is because we want to know the answer to a question. If you knew the answer about safety and immunogenicity and efficacy, then there would be no need for a trial. And therefore, there are indeed challenges in participating in trials. But this is a very, very important public good, which all of us, when able, should do that. We must not behave as if that we want the results of the trial to be administered to us, but we will not participate in the trial. That should not be the case. There has been a very pejorative term called guinea pigs being used for those who are part in a trial, and that is wrong. Every one of us who meet the standards required for a trial, particularly those who are well-to-do, who are educated, they should step up in the front. They should lead by example, so that participation in trial is a matter of public good and pride, and not something seen as, let someone else do that and we get the product.
Yes, sir. So it's absolutely safe, according to Professor VijayRaghavan, to also take part in a clinical trial. There is just no two about that. So the next question is one of the viewers who's asked us, should I get my 9-year-old daughter vaccinated? / should I get my 80-year-old father vaccinated? / can I get vaccinated if I'm pregnant or lactating? / I have diabetes. So will that have any
basically, this is an important question. So the question being asked is, vaccine trials take place in defined groups, and usually those groups are very carefully studied particularly for depending on the disease. But in the case of SARS-CoV-2, there is there will be stratification saying that the trials will not be done, for example, at this stage on pregnant women or on very old people or on very young people. And then the vaccine goes through all these trials, and the question then arises, do we then introduce them into those categories? And the reason why the answer to that is yes in this case is medical professionals estimate the value coming out of these trials compared to the potential risks of vaccinating those who are in strata which did not participate in the trials. And that is a call they take entirely on a complex set of medical grounds. What are the presentations and the risks of vaccination in a pregnant woman for this kind of a vaccine, or for a baby, or for an older person or for someone who's diabetic. So one should go for a decision taken by your healthcare professional on these kinds of matters. And I'm sure that'll be a wise and correct decision.
My next question is, why are we not able to eradicate diseases like we did with smallpox, sir?
This is a very important question. Why is it that some diseases can be eradicated and others not? And this comes from a very important quality which we need to understand about life itself. Life, all life on Earth, comes through the process of evolution by natural selection. And that means that all life is related to other life forms. And that means the viruses which infect us also infect other animals, like bats and cats and dogs and birds and so on and so forth. If a virus infects only us, then it can be eradicated. But if a virus is because of a zoonotic spill, that is, it has been there in another animal and comes, we can keep diminishing it in our context, but eventually it can spill over again. And therefore, we have to be very careful with those kinds of infections. They can never be eradicated, but there can be, their spread can be prevented because we're aware of them.
So there are several questions on your hashtag, sir. Would you like to take those, please?
I'm just trying to look at them. There was one about safety assessments and what is the status at which they are done. And I think I answered that. Safety is measured in administration and in trials across many time scales, from immediate to a few days to a few months. So the follow-up of all that is important and necessary. And therefore, it's really an ongoing process, even during the vaccine licensure. I mentioned that. Another question is about whether an inactivated virus strain is likely to be more effective against mutated strains that subunit vaccines and recombinant vaccines have. Now, and that's an interesting question. So, as I said, the inactivated virus has components on its which it presents to the body. Components which, sorry, there was some background noise. So it has components which, other than the key components which most viral proteins have, most vaccines have, which are the spike protein or the receptor binding domain and so on. So it is to the entire virus. Now, of course, when that is broken up and seen by the body, antibodies are raised to all these components. And how effective it is depends on how much those other components are important in combating the virus. Some of them may be peripheral, some of them may be important, so it depends on that, but it certainly broadens the repertoire. There are, by the way, very importantly, related to this point, while, as I said, many vaccine developers have focused on the spike protein, which is one protein, or just its receptor binding domain, there are others, and particularly in our context in India, some who have been developing vaccines against multiple components of the virus, not just one protein, but three proteins together. And therefore, that will certainly broaden the repertoire. So that was one question from which came on the.
There is a question, sir, from Shweta Kotecha. She says, what is the difference between the vaccines of the Serum Institute of India and Bharat Biotech?
That I did mention earlier, but I'll just reiterate. Sure. The Serum Institute is the AstraZeneca Oxford vaccine, which is a vectored vaccine. It uses a chimp adenovirus vector, and this vector is a standard vector used for vaccination. It's been used in many contexts, and it doesn't actually cause any infection at all. And then it puts into that the genomic information, which codes for a component of the virus, which against which we want the immune response, because that's the virus, the SARS-CoV-2 virus. Now, related to the previous question, we must also remember that when the immune response is raised against this, it is also raised against the components of the vector. So that's why, one needs to have a situation, take care that the booster dose and other kinds of steps are measured to make sure that the viral response to the protein is also there. And those steps, those work has been done. The Bharat Biotech Covaxin vaccine, in partnership with ICMR, the Indian Council for Medical Research isolated the virus and gave it to Bharat Biotech, and they grow the virus and they inactivate it. So it's completely, dead. And therefore, it's the entire virus against which the immune system works. Both of them are very, important ways of looking at vaccines. They're different. They're both, certainly going to be very strongly, standard protocols. There's nothing to be, there's nothing novel about it. The only truly novel kind of approach, which we're fascinated by all over the world, is the mRNA vaccine, which I talked about earlier. And that's, something which is coming in for the first time.
So Krishnan Narayanan writes in to say, is there any pattern from the earlier vaccines in India which can, make us discern how the public has responded to this vaccine, sir?
I get the question. There have been very major vaccination programs in India over many, many decades. The smallpox program was something which was there a long time ago. I was just remembering how, people used to come when I was a kid. They used to come home with a stove, sterilize the ring, dip it in the vaccine, and then put it on your arm. And people who in that generation had a smallpox vaccine had a ring mark on their arm, and then had two ring marks to do that. So that was a smallpox vaccine, and there was a very wide deployment of that. But I don't recall a wide, even later on, talking to people much older than me, a wide communication about it. The polio campaign was different because not only was it deployed widely, but there was an enormous campaign. And this pulse polio vaccine, the oral vaccine, had a great campaign which was kicked off from Delhi and went all over the country and resulted in oral polio vaccination. And now it has gone on to the final closure phase. Polio has been eradicated from India, but we also have preventive vaccination going on. So those are the older kinds of vaccines. The Rotavac, again, the Japanese, the Rotavac went into the childhood immunization. The maternal tetanus went to pregnant women. The measles and rubella vaccine was done in a major campaign mode recently. So there have been lots of major vaccination programs, and all of the recent ones have been combined with good communication campaigns.
So, as we speak, we've gone live on the MyGov Facebook page as well, so the viewers can tune in there as well. So we have another question from Abhay Makeshwar, and this is really very interesting. What are the precautions that we need to take after being vaccinated? I think this is a very important question, sir.
When the dry runs have been going on for the vaccination program, and they put these in mind. There are three stages of precautions, 3 stages of follow-up, which the vaccine trials and the vaccine programs look at. They look at you immediately after vaccination to see whether there is something called anaphylactic shock or not. And then, that will take, tens of minutes or so. And then they check a few hours later, you are asked to report, or a few days later, whether there are any side effects. So you can be in contact. You have 24/7 helplines. These helplines are in multiple languages. So, that shouldn't be a problem. As I said, the vaccination will not cause side effects of concern, and that is the expectation. But as you go in larger and larger numbers of people, you might have concerns, which you can call up this helpline and talk. Remember that vaccines are given to adults also, and right now the priority groups are frontline workers, healthcare workers, and people above 50. And all these categories of people will have other kinds of effects routinely, even without vaccination. They might, upon vaccination, think that some of these presentations they see are because of that. And therefore, the helpline will allow them to call up and check and be reassured that that there's nothing amiss.
So, Srivastava MM writes in to say, what is the extent of the efficacy of the present vaccine for the mutant virus, sir?
That's a very important question. But before I go to that, before I forget, I just want to answer one question which came up in the Ask the PSA hashtag, which is about why what are the manufacturing approvals given to vaccines before they were deployed. And this is very important. Normally, our regulator and regulators all over the world say that you can bulk stockpile only after vaccines are approved and not before. And what we have done this time, and this was done a few months ago, is to, at risk, at the manufacturer's risk, allow the manufacturers to stockpile vaccines should approvals come. And that is a very important step, because that allows a parallel processing. And if approvals come, those vaccines can be deployed speedily, so the manufacturing time is saved. On the other hand, if approvals didn't come, then there would be risk and at cost to the manufacturer. So it's a manufacturer's loss. So that was one question I wanted to answer. Sorry, go ahead. You had a question.
Sir, this question was what? No, we've done this question. I'll go on to the next one. I'm not sure that it's been phrased well, but it has come on your hashtag. So I'm reading it out. It's from Mr. Chandrakant. What if the vaccine immunity lasts around 8 to 12 months? Are these really enough to vaccinate all Indian citizens? I'm not sure what you're asking, Chandrakant, but I'm taking it since you sent it. Sir.
I think the question is an important one, which is, what will happen to our immunization program against the SARS-CoV-2 as we go on to subsequent years? Will the immunity last or not? Of course, the expectation is, as I said earlier, that the immunity due to the vaccines will be a significantly long. Certainly, we think over a year. How long, we don't know. But remember, this is now, once we start vaccinating and complete vaccinating a very large part of our population, there will be a range of people who have been vaccinated at different times. The earliest ones might start losing their immunity. They will be vaccinated again. And then over time, the later ones will start decreasing their immunity and they'll be vaccinated. So it's an incremental change, incremental addition, which we will see in our vaccination. It's not small, but that's something which is the system can more readily deal with without being stretched.
So the question we left out, the question we left out, I'm going back to that again. What is the extent of the efficacy of the present vaccine for the mutated virus? This comes to us from Srivastava MM on your hashtag, sir.
There is no such thing as the mutated virus. The virus which originated and had the spillover in Wuhan, China, traversed the globe, and as it went, it accumulated many, many mutations. Those mutations, by and large, are a timestamp over space of where all it has gone and what changes it occurred. And by and large, many of them didn't affect the transmission or the intensity of the disease, and therefore they are just useful to know where the virus has traveled. But some changes have been very important. The first change took place in February 2020, and that strain has essentially dominated the world. And that therefore took over the original strain with a certain increased transmission. Subsequently, there have been three events of very important concern, three kinds of mutations. One is the so-called UK strain, which I talked about, which has, as I said, even during a period of lockdown earlier, spread, and the consequences of that we're seeing in the UK. The other is a strain in South Africa, which similarly has increased transmission and also is a matter of concern. And third is a set of mutations which took place in a Danish mink colony, in an animal host, as it were, which also decreased the immune response of the minks. And that's a cause for worry, but fortunately, that didn't go out at all. In these cases, the vaccines, by looking at molecular analysis and every other tool, we think that the current repertoire of vaccines will still work against these.
Sir,
But
could
as
you
I
throw?
said, it's very important to keep in mind, we should not focus only on whether the vaccines work against these strains or not. It's very important to keep in mind that some of these strains I've talked about, the UK and the South Africa variant, seem to increase transmission enormously. Remember, I would repeat, the power of the exponential. Few people who get this strain transmit to a few others, that doubles, and soon you see a situation what has happened in the UK. And therefore, the speedy use of vaccines across the population is very important. So that benefit of a speedy deployment should be seen against the dangers of not doing so.
Correct, sir. And then that's how a whole new wave comes in, and then we found wanting because it can get out of hand. Sir, the next question that I want to ask you now is, could you throw some light on the phase 3 trials? So that's also coming on our hashtags.
Yeah, I mean, as I said, phase three trials are those where you expose people to the pathogen. But there are two categories of people in the trial: those who get the vaccine and those who don't. And let us say, for example, that you unblind this trial. It's a blinded trial, so you don't know who you're giving the vaccine to and who you're giving the placebo to. And then you find that three people in the vaccine arm showed progression of disease, and 97, let's say, in the other arm, showed, in the placebo arm, showed progression of disease. So you would say out of 100 people, 97 didn't get the disease in this arm, but and only three got it. That's the interpretation of that. So that's what a phase three trial is about. And this can be at different scales, depending on the disease and depending on the severity of the disease in the population. You might decide to do it earlier, or you might decide to unblind it earlier, or you might decide to carry it on for a longer time.
So another question that I wanted to ask you now is emergency use in clinical trials. So would you like to throw some light on that as well?
I'm sorry, emergency use authorization. What is the question?
The same. What is emergency use authorization, sir?
Yeah, so, as we discussed, vaccines go through multiple phases of trials, and typically full authorization would come after you monitor the consequences of phase three trials for some time.
Yes, sir.
Now, given the emergency now, there are multiple points at which, depending on the nature of the emergency, that you could say, I want to start deploying the vaccine even as I continue the trials. So if the disease is very, very life-threatening, you might say, I want to go right after phase one, or even before phase one, or not even do phase one. There are different kinds of diseases. For example, during the anthrax poisoning, there was a very early deployment of a potential anthrax vaccine, because it's not something which is there widely in the population, and an early introduction can protect. There are risks of deploying that vaccine amongst those who are at in danger, but the benefits are very high. If the disease is such that it's very mild, then you would let the vaccine developments go its course. If the disease covers a range of presentations, such as SARS coronavirus COVID-19 does, then you would say that because of the huge impact on a very large population and the dangers of, in that situation, of the likelihood of mutant strains arising, which can have even more severe impact, vaccination is a very important escape, and the sooner we do it, the better, and therefore we need to do it earlier. So these are the kinds of points which come up before regulators when they take a decision.
One last question, sir, on the vaccine, and then I perhaps may request you to throw some light on the way it's going to be provided to a billion plus citizens in India. We've got a very effective methodology on all of that. I read about that. So the next question is, does the vaccine work against the new strain of the virus? How quickly does it work and how long does it last against the new strain which you spoke about, sir?
As I said, the challenge of the UK strain and the South African strain, which you've referred to as the new strain, is not so much about whether the vaccine works against it or not, but the extent of its spread. And even though it doesn't seem to cause more severe disease, it seems to spread a lot faster. And that means it will affect more people, and therefore there will be more people with more severe disease. And therefore it's important to deploy all the vaccines at hand speedily against it. Are the vaccines likely to work against them? Short answer is from everything we can guess from the biology of those changes and what it means, yes, it does look like. Will there be new mutations against which vaccines may not work? We don't know that yet. And if that happens, for that, we need to be sequencing the genomes of viruses, testing them in the lab, whether what kinds of changes will cause vaccine escape, and how one can develop new vaccines. So we're simultaneously, these are not the two vaccines alone in our toolkit. We are simultaneously here developing many, many vaccines. All of that will be very useful for a variety of reasons. And we're also developing vaccines which might likely to be, not very, very likely to be effective against new variants, which the current vaccines might have escaped. So, we are putting every single approach into the toolkit so that we are protected. And that's an amazing consequence of science, that it can do this on the run. That's something we should all appreciate.
So one more question that's just come on your hashtag. About 10 million people have recovered without any vaccine or medicine. So this person is trying to ask, do Indians really need a vaccine? What would you be your take?
I would urge the questioner to go to the COVID vaccine trackers and see what devastation this has caused all over the world. It is true that most people recover, and many people don't show the symptoms of the disease also. But this is a very, very unusual virus. It infects people in a manner where many of them are asymptomatic, and therefore transmission is sort of under the radar, as it were. And then it hits some people very, very badly. The devastation it has caused is always there, and or there to see. We just have to go to different countries, look at what's happening in UK, in USA, all over India, the total number of cases, the numbers of deaths, in Africa, in Europe. I mean, these are not small numbers. Different countries have had different ratios of those who've had problems and those who have not. But the devastation has been very, very high everywhere. So we should not treat this lightly.
So does a vaccinated person also present risks to the members of the family who are not vaccinated, perhaps?
As I said, when you're vaccinated, how much of that you're protected against the progression of the disease, but how much of there is also virus in you, you're infected, of course. How much of that is shed is something on a large scale we don't know. Expectation is that shedding will be lowered substantially. But it's important that in a closed group, all those who are not vaccinated are protected, and therefore the person who's vaccinated should also take care, as should others.
Sir, if I may ask you one question, sir, would you like to throw some light about how the vaccine is going to be given to 1 billion plus, Indians? What is the methodology, sir?
So the vaccines are to be manufactured, they're done in bulk, then they go into fill and finish, then they go into distribution chains. These distribution chains start with extremely large warehouses which are appropriately refrigerated, and then they go from there in refrigerated trucks to other warehouses which are also refrigerated, and then they go from there to other locations. So this is a chain which is powered in the sense the refrigeration is there right through. And the last mile, they can go in containers where temperature is maintained, but they're not necessarily powered. This whole structure is monitored by a IT backbone called the CoWIN, and that is an amplified version of what has been used for many years in the vaccine intelligence network to monitor our childhood vaccine and maternal vaccine delivery. So this whole system has been going stress tests and tests, dry runs again and again over the last few days, and those are continuing. And this should allow correct delivery of the vaccines. Now, at the other end, at the person who's getting vaccinated, those trials involve the dummy runs, as it were, of vaccination rooms to see that you go into one place, who vaccinates it, go into the next room, who monitors, how the data is entered, is the data secure, do you know which date you are vaccinated, how do you know when your next date is, when your next appointment is, what happens if you couldn't come for the first appointment, how can you get rescheduled? All of that is also being tested and tried out.
Right, sir. So that answers all. We have yet another question, and this is very interesting, sir. I'd like to take this. As we know that different strategies have been used to formulate or synthesize a vaccine. So what approach has been used, essentially, for the development of the COVID-19 vaccine in India, sir?
Sorry, I missed that. Yeah, go ahead.
I'll rephrase it again. The question is, as we know that different strategies have been used to formulate or synthesize a vaccine, which approach has been used for the development of COVID-19 vaccine in India?
I already addressed that. There are, the two major ones I told you, but there are many others which are in progress. There is a company looking at an mRNA vaccine, another company looking at, expressing 3 different proteins, yet another company expressing a viral protein in a thermostable manner. So there are multiple other approaches. By the way, I was just pointed out, someone said that, SARS-CoV-2 is not Ebola, and therefore the emergency uses for Ebola don't match here. I should clarify. I'm not saying it is equivalent. I'm saying that for different vaccines, there are different kinds of emergency uses given. And I gave Ebola as an example of what one could do and how one could do it differently for other diseases. And for SARS-CoV-2, I pointed out the emergency comes not because of the nature of the disease. It's very complex. It covers a wide range of presentations, but because of its extraordinary spread and that continuing danger of that spread allows more mutant strains to arise, and therefore the speedier the vaccination program, the better.
Sir, another question coming in to us from on your hashtag: can a person get the COVID-19 vaccine without registration in the health department?
These are getting into
rather
questions which we will worry about a little later, if I may.
Yeah, this is more related to the admin. So I'm sure your district or at the grassroots level, wherever you are, you will get that kind of information. Anything else you'd like to say on the vaccine development of COVID-19 in India, sir? And perhaps, can we expect more such pandemics in the future, or is this the last that we're seeing? This is special.
Well, the last year has been extraordinary. It's not that potential pandemics were not discussed or thought likely to happen, but we didn't really see it happening, on a given time, such a short period. All events were to happen later on. They happen to us. Now, that said, there are very, very important lessons to be learned from this. It was not that the coronavirus pandemic was not something which was completely unknown. The intensity and the way this particular virus behaves was something which we didn't know. But we learned about it a lot in the early days, and that's quite remarkable. And that's something, first of all, we should keep in mind. The second point we should keep in mind that this is unusual. It doesn't have the severity of a hit, which allows you to focus and eliminate it in a particular location. And therefore, its main feature is this extraordinary transmissibility without causing problems to a very large number of people, but causing very serious problems to an extremely significant number. So this resulted in the breakdown of health systems. What is the way out of that? The way out of that, when you don't have a vaccine, is to have non pharmaceutical interventions of various kinds. Masks were extraordinary, and we should actually credit our system for putting that up front early. Many Asian countries also use masks a lot, and it's possible that the deployment of masks had a very major impact all over these places. So these kinds of non pharmaceutical interventions were one major tool. The other major tool, which potentially was there, was the development of drugs. Now, that didn't take off despite many early promises. That didn't take off rapidly in terms of prevention against the virus, but that was expected because viruses replicate very rapidly, and there's a small time window where drugs can act. And they use our own machinery. To get, therefore, a drug which is safe against the virus but doesn't affect us badly is a big challenge. But we did get dexamethasone, which affects the disease development rather substantially. And we learned a lot in terms of deployment of non-pharmaceutical interventions such as ventilators, oxygen, and other kinds of interventions, which was very, very important. So we learned how to get the disease down. We also learned enormously by sequencing the virus's genes to see what was happening. And that's something which has happened more and more intensely recently. And this is very important because it forewarns us about what can happen. It is not something we must keep in mind that if you have a vaccine, you can wait and you do everything for it to deploy and choose the right time. That's the perfect world. You have to balance those kinds of very difficult decisions about when to introduce with what's happening with the virus. The scale of the pandemic, by definition, it's across the world, means that there are going to be variants which have all sorts of problems. We've seen one with increased transmission. We will definitely, given enough time, if we don't do anything, see strains with other kinds of problems. Before that, we must quench the virus. How do we do that? Three things. One, we must start vaccination on scale very rapidly and widely. Second thing, remember that if you vaccinate, how many people vaccinate? If a vaccine has to be effective, then it'll be good to vaccinate a small number of people where the entire community gets immunity. For that to happen, transmission effectively has to be low. That is in our hands. So we cannot change the transmission of the virus, but we can change the effective transmission by doing the third thing, which is wearing masks and keeping distance. So even if there are mutant strains which increase transmission, if we can reduce their transmission by keeping distance, it will be very valuable. That was not done in the UK. Even though there was a lockdown, the strains spread, but there was a period where the lockdown wasn't very effective during Christmas and the New Year, and that caused serious problems. So it's very important for us to have these non-pharmaceutical interventions while we deploy the vaccine. So vaccine introduction is very important. The scale of the pandemic needs to be appreciated. The devastation it has caused needs to be appreciated. And we must therefore move now our discussions while asking all the important questions which we have. We should continue to ask them, but we should move also into a discussion on how to ensure that vaccines are widely used, accepted, and make sure that our community, our country is vaccinated so that we can reduce this disease to nothing, and also do that and help the world do that. Indian manufacturing has got a great responsibility to deliver to the world. Both companies which have got emergency use authorization have delivered to the world on multiple vaccines. Other companies are there which have also delivered. So this is a responsibility we have to not just India, but to the whole world.
Absolutely, sir. And as you rightly said, it is our genuine hope that people will emerge more conscious, not only about their health and life, but also about the environment around them and rebuild their future. So one last message for 1 billion+ Indians. They're waiting for your message. So what would your message be to India on vaccines?
Yeah, I think, this is really a very important development that vaccines have come out. But we must simultaneously keep in mind our responsibility as citizens. And they fall into three categories. All of us, when we have the opportunity, should participate in clinical trials everywhere. And for anything, for vaccines, for drugs, In this case, we're talking about vaccines. So that's a very important duty as citizens, not just here, but for people everywhere in the world. The second point is communication. It's important, of course, to ask various kinds of questions about vaccination and vaccines and their development. And that is absolutely correct. And they should be answered and will be answered. At the same time, please communicate the value of vaccination to the large public in multiple ways, whether in the media, from our science institutions, from our state governments, from our central government. This is very, very important. And the third point is, remember, the introduction of vaccines is distinct from the deployment and use of vaccines. Those take time. So aid the machinery to make sure vaccine deployment is eased. We have the process that takes time. We heard from the government today that the 30 crore vaccination of the priority groups will take 7 to 8 months. That's a significant period. And 30 crores, this has not been tried anywhere in the world. 30 crores as, in a population of 130 crores. And this is a huge effort. So we must all appreciate that it takes time to deploy even into this priority group and then into others. So we must be patient and follow all measures. And that's very, very important.
And on that note, sir, many thanks for joining us in this very, very interesting conversation. I thank you on behalf of the entire team of India Science. And thank you for sharing your wonderful insights. And we hope that we will have more such conversations in future. With this, viewers, it's a wrap on this edition of The Science Behind Vaccines. I thank you all for watching. Namaskar.
Namaskar. Thank you very much. I just add one point that if there are any questions which we may not have answered, I'll be certainly happy to take them separately and get back to them on social media. Thank you.
Thank you, sir. Namaste.
Namaste.